All right, thanks very much to the Allen Foundation for the opportunity to be here, and especially for the opportunity to be part of this new partnership between the Allen Foundation and the American Heart Association. So I study heart mechanics, and that means when I picture myocytes, I picture cells that look something like that. Aligned, elongated, with a very highly organized structure of contractile units called sarcomeres. But when we take cardiac myocytes out and try and study them in culture, instead we get cells that look like this. And so it's taken some time to try and figure out how to make cells in culture look more like the cells that we were trying to study in the first place. And there are a couple of ways to do that. One, which is shown here, is to micropattern extracellular matrix onto a 2D substrate so that you restrict where the cells can attach. Another one is to actually encapsulate the myocytes in a mixture of extracellular matrix proteins and then electrically and mechanically stimulate that construct and develop it over some time to get something that actually starts to look a lot like how they looked in vivo. What's interesting to me, though, is how hard this turned out to be. This took a couple of decades of cardiac tissue engineering to really be able to do this well. So contrast that with a different experiment. This is from Doris Taylor's lab, where, again, they took a heart, and this is a different stain and zoomed out a little bit, but these are those aligned cardiac myocytes. They perfused that heart with detergents to get rid of the cells, and so what's left is all the empty spaces, if you're a myocyte person, or extracellular matrix, if you're me and Sunil, that surrounds all of those cells in the heart. Then they took cultured endothelial cells. These are the cells that line the inner walls of blood vessels, and they perfused them back into the scaffold. And the endothelial cells repopulated the insides of the vessels in the scaffold. And they took myocyte-like cells, this is a cell line, perfused those into the scaffold, and the myocytes repopulated the spaces, the cells repopulated the spaces where the myocytes used to be, and they aligned and they started forming these organized sarcomeres again. So these tissue engineering examples, I think, suggest something that we're learning about the cardiac extracellular matrix, which is that a lot more than we realized of the phenotype of those myocytes that we're interested in is due to instructions from the surrounding extracellular matrix. And that's turning out to be true not just of a structural scaffold that it gives them places to attach, but also that attaching to the different extracellular matrix proteins is providing critical signaling instructions to the cell that regulate its phenotype, that growth factors that are bound to the extracellular matrix are locally active at the cell surfaces, and even that fragments of the extracellular matrix proteins can break off and start to serve signaling functions in communicating with the cells. So there's one more thing that's really interesting about this whole story if you study heart disease, and that is that the half-life of the proteins inside the myocytes is on the order of a few days, maybe a week. But the half-life of many of these extracellular matrix proteins is months, years, or in some cases even decades. And these matrix proteins accumulate modifications over time from aging, from inflammation, from diabetes, which maybe not coincidentally are also all the main contributors to heart disease. And so the premise of this project is that in order to understand things like heart disease and aging that develop over the space of decades, that we need to understand these dynamic cell matrix interactions and particularly understand how cell or other modifications to the matrix can change the instructions that later cells are receiving. All right, so the challenge here is that we have a bunch of tools for making measurements about extracellular matrix, and each one of them gives us a snapshot, a small part of the story, but not everything that we'd like to know. So, for example, we can look at the composition of the extracellular matrix using proteomics and how that's changing over time. In this example, they're doing that after a myocardial infarction or heart attack. We can use staining and microscopy to look at how a particular component of that extracellular matrix is organized spatially. And in this case, I'm showing you stained large collagen fibers in scar tissue that forms after a heart attack in a number of different animal models. Or we can use labeling techniques to try and assess the turnover of a particular matrix molecule that we're interested in. This is work from Michael Kjær's group in Copenhagen, where I had a chance to do a sabbatical, and they're looking at turnover of collagen in human tendon. And what they find is that those turnover rates are fractions of a percent per hour. It works out to about 1% per day. And those rates can go up quite a bit after a bout of intense exercise, like running a marathon, which is what they had their volunteers do in this study. So with all these tools at our disposal, we know a lot, but there are still some really fundamental puzzles that we have not been able to resolve. And here's one example. So this is also Michael Kjær's group in Copenhagen, so same group that did the turnover measurements I showed you a minute ago. And here what they're doing, this is the curve of radioactive carbon in the atmosphere due to above ground nuclear testing. And as you see, since we stopped doing that in the 60s, there's basically a steady exponential decline. And they measured the levels of that radioactive carbon in biopsy samples that they took from tendons in patients at a bunch of different ages. And what they're showing here is their fit to that data. And basically what they concluded from their fit to that data is that essentially all of the collagen in almost every tendon they sampled, except for those guys, was deposited before the person was 17, even in patients that are in their 70s. So how can both of those things be true? The collagen's turning over at 1% per day in the tendon, but at the same time, all of the collagen that's in the tendon was made before you were 17. Right? One possible explanation for that is that the turnover that they're measuring with the first technique is highly spatially localized. It's only happening right at cell matrix interfaces and not getting incorporated broadly into the tendon. But this is where our current measurement techniques start to fail us. We can't really see that. So one way to get around that problem a little bit is to use computational modeling. So these are some models that my group uses where we're showing fibroblasts migrating into a healing myocardial infarct, depositing collagen, and changing the alignment of that collagen over time. And we've used some models like this to try and integrate snapshots of information that we get from these different techniques. In this case, integrating snapshots about composition, about structure, and about turnover at certain points in time to try and explain how patterns of collagen alignment that we observe in healing scar tissue emerge. We can go a step further, and for each one of these cells that's crawling around in this model, we can represent the signaling pathways so that we can start to represent what we know about how attaching to different extracellular matrix proteins or interacting with those growth factors and cytokines I told you about affect the behavior of that cell, including how it modifies the surrounding extracellular matrix so that the next cell that comes along sees a different matrix. But that still gets us only so far, and so we really think that something that's important is that we need techniques that can visualize when and where cells are modifying the extracellular matrix, and then how those modifications affect what the next cell is able to access in terms of the stored information. And so to do that, we're adopting a technique called STOMP, and this basically allows us to target, pull out, and extract for analysis any region that we can define by any kind of staining technique that we want to use, the antibody staining technique. So for example, this little diagram I showed here, these little red dots are, suppose we had a cell surface marker and we wanted to stain and label those cell surfaces and then pull out all the surrounding extracellular matrix that's right around that cell surface. We can combine that with labeling techniques and ask, how is the matrix turning over right at the surface of that particular cell type? Or another example, we could pull out, let's say, the large collagen fibers and ask, what are all the things that are bound to those fibers, and how is that cocktail turning over and changing over time? And we're planning to do this with aging and then with healing after myocardial infarction in both young and old animals. So this is the project team. You've met me. Sarah is an immunologist who is our expert in the STOMP technique. Eli is interested primarily in stem cells, but is also working with mass cytometry and cell barcoding that some of you are probably familiar with. Sasha Klebanoff makes targeted liposomes and microbubbles for drug delivery, for imaging, and in this case, we're using some of those tools to try and read out how matrix modifications change the accessibility of particular binding sites for the cells. And then there are a number of collaborators and contributors listed down here that have helped us in planning this project and in thinking about this problem of extracellular matrix turnover and information storage. Thank you.
So the use of microsampling for proteomics, non-optical but mass spectrometry, there's this fellow in Germany who has this picosecond IR laser that can just sort of blast cold, you know this?
Yep.
I forgot the acronym, but are you going to try any really local information, you know, extracting proteoforms from a very local part in your tissue?
Yeah, so there are a bunch of these techniques, and I get lost in the acronyms too, because they all have different acronyms. But I think the person you're thinking of might have been Bernd Bodenmiller, who was one of our consultants on this. So I think the main difference is that a lot of the laser ablation techniques are going along and ablating locations, right? So each time you pull a bunch of stuff out and send it to the mass spec, you know that it came from a certain location. What we like about the optical one is that you first get the map of where you're going to pull, and you can target, for example, the one I really like is that cell surface example. So target the region around particular cell types and pull those out. So it's a variant on the same technique, but I would say maybe conceptually it's a little bit more like an extracellular matrix version of laser capture dissection that people do when they're looking at RNA, if that makes sense.
It's easier to register because it's the same, the images.
Yes.
You act right on the images right there.
Well, in fact, what you do is you do the confocal image first, and the confocal image becomes the mask to go back in and photo cross-link to pull the stuff out. So you always have the image of where everything was before you pull it out for analysis.
And the kinds of modifications on the proteins as they age, the ECM, I mean, yes.
Yes, would you like to collaborate? How long exactly is the list? Yeah, I mean, obviously the really low-hanging fruit ones for diabetes, for example, are the glycations, right, the cross-linking, but there are so many, and it's a little bit difficult to decide where to start.
It's kind of like the
eye. I'd love to talk to you
way.
about that.
Right,
yeah.
sure, okay.
Okay, thank you.